Well, I thank the organizer for the invitation. It's really an honor to be able to present the talk. This turned out to be a very good complement of Partha's talk. And I make the title rather general, Big Data and Cancers. So the first part of the study of this talk is to summarize what has happened in the last decade. I'm writing an annual review, genetics review, so it's a good chance to go over that. So what I think about big data, there are two kinds. The first kind is that in the past, people have collected a lot of genome data for specific purposes. And so in those cases, NCBI, for example, compile all the data, and then the big data are very useful in that respect because the small data were created for some particular reason. But then there's what we call type 2 data. This is the creation of large data set to solve a defined set of problems. And human genome is one, and the cancer genomics I would be talking about is another one. And this is really something we have to think about. So the TCG, the cancer genome data, really asks the question whether tumorigenesis is a process of convergence. That tumor development follow a set of pathways, and we just have to understand those pathways. So in other words, the question is that, does the same cancer type usually have the same set of mutations? If they do, we'll try to find them. And so the background in cancer, I think Partha has talked about this, I can skip this, that it's a genomic disease, and the large, long-lived animals like us are particularly susceptible to this. So we live long, we have 10 to 13 to 14 cells, and anytime any cell acquires some mutations and out of control, then we are in trouble. And that's why mice do not have to worry about getting cancer. We do. And so in this backdrop, the Cancer Genome Atlas was launched. And now they were about more than 10,000 cancer genomes sequenced, and against the normal type. And they are done across more than 30 cancer types. And by now, over 500,000 coding mutations have been obtained. So then we can see what we can learn about this large data set. So you look at this, that the number of cases, these are the more than 30 cancer types, and the number of cases hover about 200 to 400 cases. So these are not small data. But the result turned out to be somewhat unexpected. So in this, each column is a cancer type. Each row is a mutation. You don't have to read this. It's just a huge compilation. The message in the end is in heading that P53, as we know, occur in 42% of all cancers. The second one is PIK3CA, occurred in 18% of all cancer patients. Surprisingly, there's no third one that account for more than 10% of cancers. So the very original hypothesis that there's a certain pathway turned out not to be true. Cancer seems to be, everyone seems to be different in a way. The pattern is not there. And then you can try to see that what I talk about was across all cancer type. And again, this is genes, and these are cancer cases. You can see the top two, P53 and PIK3CA, they really account for different types because they are so much more common. Occasionally, you find some genes that are particularly clustered for kidney cancer, VHL, for example, some gene cluster for breast cancer, but you can see, by and large, the plots are scattered. A few of them concentrate in certain gene, a few. Only two that apply across, but then what? The commonality sort of disappear. And so this is an analysis of identifying the cancer driver gene by definition. It's not an evolutionary term. It means that genes whose mutations are overrepresented in cancer types. Okay, so there are about 10 to 35 genes that are overrepresented either in most cancers or in some cancers, about 10 to 35. Now, then we come back to this issue that I think it's an important issue that should be addressed. That is, when you have very large data set, then you can see very small differences. So the question is how biologically significant are those statistically significant results? So in other words, there's something I call the paradox of more begets this. You take an example that suppose you want to look at sex ratio of human population. If your sample size is 1 million, then you can detect a difference of 50.1% versus 49.9%. And you can really show statistically they are significant, but it's the result really biologically significant. It's the results robust. And that is what I think is a paradox of too much data, so more actually begets less. And so the reason I talk about this is that in cancer genome data, you can see that because of the huge funding, large data set, sometimes people tell you a p-value, 0.001. And you say, just exactly how does that 0.001 translate in magnitude? So one way you look at it is that we did this analysis by just looking at the enrichment. In other words, if you compare the occurrence of mutation across the population and in the diseased patient, you ask the question, how much enrichment is the mutation in the disease? Is it 10-fold, 100-fold? And here is, in breast cancer, the x-axis is the number of mutations in the case of cancer, and the y-axis enrichment. So they are the top 5 genes. The top 5 genes usually have an enrichment of 10 to 30 times. In other words, that the disease patient, the probability that the patient having the mutation is about 10 to 30-fold more than the population. But you can see there are only 5 genes, and then there are about 100 genes that has an enrichment of about 3-fold. Okay, not very much. And there's another thing that bothers me about this particular trend, because supposedly if the patient had fewer mutations, the enrichment should increase, but they don't. And so the result comes from the individual gene analysis is not really a very small number of genes upon mutated increase the cancer risk. Okay, the enrichment can be interpreted of increase in the risk. That's if you have the mutation, the chance of lifetime cancer is how many fold greater than the rest. And the bulk mutation would increase the risk by less than 3-fold. And so that's really what the cancer genome have told us, that it's really not, the results are not what people expected. So then one move on to say, well, perhaps it's not individual genes that are responsible, and I think Partha extensively covered this. And this question is that maybe some pathways are more enriched. And then you can classify these genes by, so there are studies that start looking at the rare somatic mutation and see what combination may increase the chance of getting cancer. And I don't go through, you can see that they identify subpathway and so on. You can see that you look at these motifs and see where they are enriched. The choice of these motifs in the first place is somewhat questionable. But anyway, the study did identify some genes that in combination increase cancer incidence, and I would not go through this. But here's what the question, that people try to identify mutation combination that drive cancer. Now, if you can see that if the disease patient has 10% chance of having a particular gene being mutated, and then you are looking at a combination of 3, you are looking at about 0.1% of cancer patients. And if there is a 10-fold enrichment, this is all very optimistic. estimate that you are looking at 1% of, say, liver cancer patients. So again, this is the paradox of large genome, that no matter what happened, you can always see some signature, some signal, even very weak. But because the data set is very large, they always give you a very big statistical value. And so this sort of logic can be pushed on, and then you can look at different cancer types and identify combination of mutations. And then Partha allude to this, that then you can say, well, perhaps we can classify 12 cancer types. In this case, the classification may not be aligned with the pathological or the tissue of origin. Maybe different cancer types from different tissue would share the combination, or the same, I think in glioma or something, that it actually has 3 types. I don't remember the detail, but you can see this attempt, this sort of data mining. You try to see certain pattern that well, maybe liver cancer has a certain profile, breast cancer has 2 different profiles, and so on, and this sort of classification. But then you can see when you start doing this, for example, people would say colorectal cancer has certain type, but if you dig in, you can actually find that you can see 4 more types within the colorectal types. And then people start putting expression data into the analysis, but to try to extract different types out of it. But let me, before I get down to the conclusion that we did find some other problems in this study in Beijing Institute of Genomics. That is, if you look at different institutes, like for example, St. Louis, Washington U, and Broad Institute, and Santa Cruz, and they all identify mutations in the same patient, but the different centers actually call mutation very differently. The overlap is between 50 to 70%. But if you look at, and then you can see this is just a table, and this is the tools we use that develop. By the way, that there are many, when data are so large that slight difference in the tools will give you very different results. So TCGA has a competition with simulated data and so on, and people try to compete. And one of my associates is very keen in doing this, and got a gold medal and silver medal in this sort of. But here, when you use all these tools, you find that especially when the mutations are not in high frequency in the sample, the overlap is very, very low, okay? And there are many mutations that are, well, so when you say, well, the patient has EGFR, then most doctors actually tell you that some cells of the patient has EGFR mutations. But what you need is that every cell has this EGFR mutation. Otherwise, you just kill 20% or 50% or 90% of the cells. But nowadays, there are treatment regimens that as long as the patient has this mutation in some of the cancer cells, they will use the drug. And when you talk to the doctor, you say, wait a minute, only 10% of this guy's cell has this mutation. Why do you use this drug? And say, well, that's what the rules are. And you say, well, you kill 10% of cells, well, then what's going to happen? Well, then that's something else you have to deal with. And so you can see there are a lot of low-frequency mutations, and the prediction is very, very different. And the reason they are low-frequency mutations is something I want to address. That's tumor within tumor heterogeneity. So let me summarize this, that the whole big data were created with the underlying assumption that tumorigenesis represents a small set collection of developmental programs. So we have a good chance of identifying the mutations, and we can understand how tumor developed. But the alternative hypothesis that an evolutionary biologist would tend to subscribe to this sort of view, that the cancer really is a process of divergence. It's the multicellular organism, the cell return to unicellularity, and there are very large number of mechanisms that to become cancerous. And the truth certainly would be somewhere in between. But is it closer to the original assumption of developmental programs gone already, or it's more the view of evolutionists that there are many, many ways of getting to the same thing? This is not unlike the thinking about organisms invading Tibet Plateau, cattle, pigs, wolves, human, going up to the plateau. I mean, how many ways, how many genetic mechanisms you can use to become adapted to that. And it's actually very, very important, because when one spends so much money and effort, the underpinning hypothesis has to be rigorously examined. Anyway, so the part two, that suppose cancer development is not really development, it's just evolution, then should we not look at each cancer case instead of looking at 10,000 cases? Why don't we do what population genetics do, that we study human population, only one species, in great detail, and try to understand the evolution history of humans? And you can say, well, perhaps the evolutionary history and mechanism of humans would not be the same as pigs or cattle. But the view is that if you understand something very, very deeply, you would have better idea how to apply it, rather than looking at 10,000 cases and in each case only casually. So we decide to do that, and we look at a case very, very in great detail. And the paper has just been published. You can see that Wensheng and I co-authored the paper. And in this case, we really treat a single tumor as though we are studying human populations. And so you take samples from Africa, from Europe, from Asia, from Americas, and trying to see how tumor grow and cell migrate and so on. So we want to know how many clones are there in a single tumor, just one tumor. And what's the size distribution? So you take a tumor, you take a slice, and you sample 300 sections. So this is how we sample it. It doesn't look pretty, but I can somehow, it's nicer to look at this. So this is a slice of tumor, 1 millimeter thick, 3.5 centimeter across. And each point is a sample, and we sequence or genotype everything. The red dots are sampled. The blank dots are genotype. And you can see this clonal structure, okay? From middle, it follows a geography. You can see this more clearly. This is the map of a tumor. And this looks like Africa. This looks like China. This looks like Europe, America. And there's a map. And you can see the growth pattern. And each star represents a singleton. So now we have an entire history of evolution of one particular tumor. And then we can really estimate the divergence using population genetic theory. I don't want to go into the detail. I mean, people who study population genetics know that we look at frequency spectrum. In other words, we look at the number of mutations and look at the frequency of the mutations in the tumor. And by combining the information, we'll know whether these clones evolve by natural selection or by random genetic drift. And since the population is expanding, the theory is actually somewhat more complicated, a lot more complicated than the conventional population genetic theory of constant population size. A lot of these were done theory by Rick Durek of Duke University. But anyway, this is the upshot. If you understand this, then you can estimate the total number of coding region mutations in this particular tumor. Okay, you can look at your sample and extrapolate. The minimum, the more conservative estimate of this is about this. And so in other words, that this particular tumor has about 100 million coding mutations, 100 million. And the mutation rate of this tumor is unexceptional. It's not surprising because the tumor has more than 1 billion cells. So that it has 100 million mutations is not that surprising. And you can see that by doing computer simulation using the parameters, you can see the clonal structure. And here, a clone has to have 50,000 cells to be shown different color. Then you take a small region, you blow it up with the clone size being 5,000 cells. Then you take another piece and you blow up with 500 cells. And this is really, you can see it's really like a kaleidoscope, that it's full of mutation, full of variation. And so the point is that using the example that when we collect big data to address specific scientific problems, we should delineate the hypothesis very clearly. And when we look at tumorigenesis, we think that it's really should be modeled as an evolution phenomenon. It just happens to have medical significance. So I'm coming back to this oversighted dictum in evolution biology. Although it's said repeatedly, it's in fact correct. Just like you say that one should be kind to one's neighbor. I mean, it's a correct statement, even though it had been spoken too frequently. So the work is done mainly at Beijing Institute of Genomics, and I travel between University of Chicago and Beijing, and its funding source. Thank you very much.